Okay, welcome back everybody to the Recombination romp. We're going to continue on. Our next speaker actually has a pre-recorded video here, so I'm going to press play, or attempt to do so. And hopefully we'll hear them. Or not.
I'm a postdoc with Grace Lee.
Okay, it's a little quiet.
The symposium organizers for this opportunity to present our work. Meiotic recombination is a fundamental process across sexually reproducing organisms.
Is that going to be too quiet?
Interestingly, the rate of recombination varies by about 3 orders of magnitude across taxa. It also varies across individuals within species. Here's an example highlighting such variation in humans. Recombination rates also varies across genomes. Here is a recombination map for the second chromosome of Drosophila. A prominent pattern is a big dip in the middle, thus heterochromatin. Heterochromatin is a repeat-rich gene pool region. It is also enriched with repressive marks such as H3K9 di- and trimethyl, DNA methylation. Why is recombination suppressed in this region? Here's one possibility. Studies in Rhabdopsis found that the enrichment of repressive marks at heterochromatin is negatively associated with spawn 11 binding, which generates double-strand breaks. And double-strand breaks is the first step for recombination to happen. So that's a broad genomic pattern. How about the finer scales of variation in euchromatin? Euchromatin contains most of the actively transcribed genes. In addition, it harbors many transposable elements, TEs. TEs are selfish elements that can replicate and jump. Sometimes they jump into genes and disrupt gene functions. To suppress the selfish behavior of TEs, hosts can epigenetically silence them by deposition of repressive marks, so they cannot express and jump. Interestingly, these repressive marks could spread for more than 20 kb, forming little heterochromatin islands in the euchromatic genome. So because the repressive mark is negatively associated with the occurrence of double-strand break, the first step of recombination, and euchromatin TEs can form heterochromatin islands. We hypothesize that the epigenetic silencing of TEs reduce local recombination rates in euchromatin. To test the hypothesis, we need to efficiently identify recombination events. First, we could cross 2 inbred lines to generate F1 where recombination happened. Then backcross them to F2s. The standard approach is to genotype every F2 individual. To find a large number of recombination events, one needs to genotype many F2s. But we think that if all we need is haplotype information, can we simply pool all the F2 individuals and sequencing them with long reads, which contain haplotype information? So we generate benchmark data to test this idea. We cross 2 Drosophila inbred strains that have been fully sequenced with PacBio long reads. And we collect about 200 F2 individuals DNA will extract from each individual. Half of DNA will sequence individually with Illumina. The other half will pull together to sequence with PacBio CLR. In these studies, we only focus on crossovers, one of the two recombination products. For each long read, if the parental origin of SNPs switch, we call it a crossover event. It can be narrowed down to less than 1 kb, depending on SNP density. So compared to the Illumina benchmark data, our new approach has relatively low false positive rate, but high false negative rate, more than 60%. Simulation of PacBio long reads confirm the observation. So further investigation suggests that the high false negative rate is partially due to the stringent criteria we use to exclude potential gene conversion events. And the false negative events will randomly distribute, not biased to particular genomic regions. So if one wants to detect more crossover with the same precision, they can increase the number of individuals in the pool and sequence it to higher depth. So we apply the method to two large pools from two crosses, A6 and A7 cross to A4. Each F2 pool has about 7,000 individuals. And we detect about 1,400 crossovers from each crosses. Based on the back of envelope calculation, the recovery rate is about 65%. Next, we compare the crossover distribution along the genome in our recombination map with an existing map generated by the standard genotyping approach. The correlation between the two maps is 53%. It's worth noting that in the previous studies, the average correlation in recombination maps between two strands was about 45%. This suggests that our methods recapitulates some of the known genome-wide variation. So now we want to test whether TEs has negative impact on crossover. First, for each strand, we select 5 kb flanking windows of TEs. We also select control windows that are far from TEs. And we observe that windows with TEs have fewer crossover events than the controls. Then we ask whether this reduction is due to TE-related repressive marks. We estimate the enrichment of a repressive mark, H3K9 trimethod. We call it K9 enrichment around TEs. And we correlate that with the number of crossovers in these windows. There's a strong negative correlation in one of the two strands. However, these are within strand comparisons, so different genomic regions may have different features affecting crossovers. So to control for that, a more direct test is to compare the homolog windows with and without TEs between strands. So we first perform downsampling to control for different read depth between homolog windows. Then we calculate the differences in crossover numbers between them. If the differences are lower, it suggests that the allele with TEs have fewer crossovers. As control, we select window pairs that far from TEs and calculate such differences in crossover number. First, we consider Ts without K9 enrichment. In one of the two comparisons, the differences in crossover number are lower for Te-related windows than the controls. Next, for the Ts with K9 enrichment, the differences in crossover numbers were lower and were significantly lower than the control windows in both comparisons, supporting our hypothesis that Te-related K9 enrichment reduce crossover. So our genomic analysis suggests that Te has negative impact on crossover. Now we want to use an orthogonal approach to experimentally test the effects of candidate Ts. We generate strands with Te constructs, about 7 centimorgans from a reasonable marker back, and conduct similar crosses. The crossover rate was estimated using the allele frequency decay approach. We select F2s for homozygote BRAC and pool them for illuminal sequencing. The allele frequency decay from the BRAC markers can be used to infer crossover rates, and we focus on that around the Te constructs. So we have strands with different constructs insert at the same place, a control without Ts, a DNA-based Te 3060, and 2 RNA-based Te, U, and cobia. We observe a trend that the RNA-based Tes show reduced crossover rate relative to the control, consistent with our hypothesis, because RNA-based Ts tend to show higher K9 enrichment than other Ts. So to summarize, we developed a novel approach that uses long-read sequencing to identify crossover events among pooled recombinants. And we found that the presence of Te reduced local recombination rates, contributing to a different recombination landscape between individuals. The effects is largely driven by Ts with enrichment of repressive marks. So with Te contents diversified across populations and species, Ts could be contributors to varying recombination rates across taxa. With that, I want to thank my lab mates and collaborators and colleagues for discussions. And thank you so much.
Right. Hi. Thanks everyone for being here after lunch, and thanks to the organizers for inviting me, having the opportunity to present for the first time in evolution. I'm very excited about that, and I'll be talking about what we are doing at the Pfeiffer's group at Arizona State University. We are investigating crossover and non-crossover recombination events in AIs. It's a species of lemur, Daubentonia madagascariensis. So I had like 7 slides of presentation, but I got rid of them because probably if you're here, you already know what Meyer recombination is. But in any case, I would like to stress the fact that it's very important from a molecular perspective because it prevents aneuploidy and ensures that the chromosomes are segregated during gametogenesis. And also it's very important from a evolutionary perspective because it allows alleles to shuffle and purge deleterious alleles. New combinations are also possible, and so far we've been mainly talking about crossover events, but recombination can be resolved in 2 different ways, both recombination, crossover and non-crossover. First of all, recombination is generated by this DNA double-strand break, and depending on how it's repaired, we can have a crossover event or a non-crossover. Normally, crossovers are large sections that are exchange between homologous chromosomes, while non-crossover events, it's a non-reciprocal transfer of smaller part of the DNA, and it's only happening in one of the chromosomes. So variation has been widely described and happens a lot. So recombination is very variable depending on the same individual, so depending on the chromosome. Also within the chromosome, and there are some regions, the hotspots, where the frequency is much higher. Also within the same species, there are different variation between individuals and between females and males and so. But here I'm more focused today in the differences between species. And in that figure, you can see that the loci MDH is the recombination there. It changes very much from species to species. And what we are doing at the Pfeiffer's group, it's more focusing on non-human primates. And here, this shows that the recombination changes very much within the primates, depending on if it's a human, bonobo, chimpanzee, gorilla, and so. So that's not something new. I'm not talking about it. So it's been widely described for more than 10 years. Recombination rates have been checked and compared humans, chimps, bonobos, and so, and that has been later confirmed with vervet. This is the gold variation, as you can see, is much higher than humans. But what we have something new now, which is that because of the availability of sequencing data, now we can check very carefully both recombination events like crossover and non-crossover. That was already a few years ago in the baboon colony, and my colleague Cyril also worked with rhesus macaque. But the species I've mentioned so far, they are all at the very end of the phylogenetic tree. And mainly we're talking about all wool monkeys. And if there is already a lot of variation in all wool monkeys, we wonder what happened if we check species that are early diverged. That's why we focused on the lemurs. And particularly the species that we are focusing on is the ai, this very cute animal that lives in Madagascar. And it has several particularities. One of them is that it uses this very long finger to scoop the food from inside the trees. And then there are like only 2 species of trees that this animal is able to feed from. Hence, the distribution of the animal is very, very, very large. It's the lemur with more range, and that's why the population densities are very low. And because of that, it's endangered. And we wanted to know whether, by understanding the patterns of recombination, we could understand a bit how to help conservation planning. For that, we obtain whole genome sequencing of 14 individuals. That's around 35-fold whole genome sequencing, and they are organized. This is a very intricate organization, but we have 3 multi-sibling families, and those are also organized in 6 different pedigrees. And I'm putting them separately because depending on how I deal with the data, the crossover and non-crossovers are differently identified. First of all, to identify crossover and non-crossover events, what we need is phase informative sites. So we need to understand what SNPs are coming from the maternal side, or which ones are coming from the paternal side. And because of that, it's important to phase the genotypes. With a pedigree data, you can do that by checking when you have a heterozygous allele, and then you have parents that are different genotypes, and you have either a partner of offspring that is homozygous, then you can trace 3 generations the phase informative markers. And if you realize that instead of coming from the grandmaternal side, it's suddenly coming from the grandpaternal side, that's a change of phase. That's how we can detect change of phase with pedigrees. With families, it's a bit different because we don't have the parents, let's say. So what we need to do is, normally the information that it's from the F1, we don't have where the SNP is coming, whether from one haplotype or the other. So we take one of the offspring as a template. We assume that it's inherited from either the paternal haplotype or maternal one, and we compare all the children, all the offspring with this one. And when we find, for instance, a 3rd and the 4th case, when we find that there's something that should not be there, we understand that that's a phase of change, the change of phase. So when we have the phase informative markers, then we can detect the CO events, which is like longer than the NCOs, but we need to take into account that maybe if they are towards the end of the chromosomes, or maybe if they are too close, that could be a false positive, so we remove them. And for the NCO, we do the same. We also check the distance between them. We check if they are close from the ends, and find also, because they are normally 1 or 2 or 3 phase informative markers when it comes to non-crossover events, we need to check also if they are part of a like polymorphic region, and then we also get rid of them. That's the methodology. What we found is mainly 147 crossover events, which are distributed in the different scaffolds, separated in male or female. Those are very preliminary results. I was working on that a couple of weeks ago, so no time for me to really process the results. But what we found is that they are clustered, as we would be expecting, that in some chromosomes there are more, in some scaffolds there are more events than others, and male tend to be a bit more closer from the telomeres than females, but yeah, that's a work in progress. Something interesting is that unlike other studies that I mentioned before, the resolution is lower, and that's because we're working with whole genome sequencing data instead of array data, and that allows us to have a very fine resolution. Resolution implies that the distance between the previous phase informative site and the first after the change, it's shorter, so we can actually determine quite well where this crossover event occurred. I was probably expecting a bigger screen, but in any case, what I'm plotting there is a genetic map, and what we see that it's also not surprising is that the shorter chromosomes have a higher recombination rate, and the longer chromosomes have a larger one. For the NCOs, I must say that normally, because of the tiny sections that they are part of, we tend to underestimate the number of NCOs. So we are confident with the results, but also it's possible that we are actually missing a lot of them. And the NCO track, it has a mean of 30 base pairs. It means that normally when it's longer than one phase informative site, it can take up to 30 base pairs. So that's indeed a very short distance. I forgot to mention that we are studying the same from both perspectives, from the pedigrees and from the families. That's why I separated that. The results from the families are still ongoing, so I will only introduce the COs. We have many more crossover events because there are many more meiosis, but more or less the mean is similar, and the resolution is kind of similar. It's a bit packed, but again, we can see that, for instance, scaffold, I think it's 16, it's very clustered in one side, and that's more or less what we expect to see. And again, the genetic map is showing that shorter chromosomes have more concentrated, more like higher recombination rates. I hope I can follow up in the future, providing more results, explaining how that compare to other primates, also to see if it can be further compared. And then I wanted to do the same with an linkage disequilibrium-based approach, see if the results match. That's what we expect, but it would be nice. Then we generated a very nice and accurate reference genome for IIs and also the annotation. So now that we have the annotation, we can actually check if there is an overlap in the hotspot or the region we detected with some functional elements. And we will, with this same data set, estimate the mutation rates to complement the study. Obviously, I'm not doing that alone. There is a great group that are working and helping me and actually doing a lot of work. So they will be presenting, Tarbot is presenting later today in the population genetics session. And Cyril will be presenting a mutation tomorrow. So if you are already in love with these cute animals, or if you are interested in how my results complement with them, feel free to attend. It will be super interesting, I'm sure. Finally, thanks to my supervisor, Suzanne Pfeiffer, in the Pfeiffer Lab, Jensen Lab, which we collaborate a lot. We do a lot of computational biology, so we need to import cluster that works, so thanks for the research computing as well. And if you are interested in recombination rates in non-human primates and so, we are looking for postdocs, so reach out, and I'm sure you will have a great time there. So thanks for your attention, and I'm taking any question now.
Okay, so hello everyone, and so today I will present you a chapter of my PhD that I recently defended about the role of the PRDM protein in the regulation of recombination events in salmonid species. So, as you know, recombination is a very important process, both from a mechanistic and an evolutionary point of view, because first, it is essential for the proper segregation of the homologous chromosome in Asia cell during meiosis, and it allows the genetic mixing that generates new combinations of alleles. Recombination rates are variable along the genome, and their variation forms what we call recombination landscapes. And these landscapes vary at different scales. They can vary at a very fine scale in the kilobase order, where recombination events tend to concentrate into small regions of about 2 kb, called recombination hotspots. And they also vary at broader scale on the megabase order, where large regions can be characterized by higher recombination rates. And today's two main mechanisms that regulate recombinations have been identified in animals. So in many species, such as in birds, canids, some terrestrial fishes, recombination events tend to be located in open chromatin regions, such as in promoters, and especially at transcription start sites, TSS, and also CPG islands. And in these species, the recombination hotspots are highly conserved over large evolutionary timescale. So, for example, here we can see in birds that the recombination rates of the green bird species is significantly increased at the hotspots of the orange bird species. On the contrary, in most mammals, such as in primates and mice, recombination events tend to be localized independently from the distance to the nearest promoter-like and CPG islands. And in this species, the recombination landscapes evolve very rapidly. For example, here in the chimpanzee, the recombination rates in green don't increase at the location of human hotspots, which means that they don't share their hotspots. And in this species, the recombination events are regulated by the PRDM9 protein, which binds to a specific DNA motif to recruit the recombination machinery. And different PRDM alleles target different DNA motifs. In the first group, characterized by stable hotspots, this PRDM9 protein has been lost or is no longer functional, and we talk about the default system. And this system is usually opposed to the PRDM9 dependent system with stable hotspots. And these two types of landscapes are not quite well described in these species. However, little is known about the recombination landscapes in other animals, and especially the role of the PRDM protein. So here is represented the phylogenetic distribution of PRDM9 in animals, and we can see a mosaic of presence of the protein in green and absence in red. The identification of full-length PRDM copy in Clindian suggests that PRDM ortholog was present in the common ancestor of animals, but multiple losses have been documented in several lineages. And whether the protein is present is a preliminary step that has been investigated in many species. However, the role of these copies of this PRDM ortholog in non-marine species is still unknown. And so the question of its role in non-marine species arises. Does this PRDM copy play the same essential function in the regulation of recombination than in mammals, and thus being an ancestral function, or is it playing another function, and the function in recombination only appearing in mammalian species? Among teleost species, a full-length PRDM9 ortholog have been found in some salmon fishes. And so during my PhD, I investigated the recombination landscapes in this group in order to assess the function of PRDM9 in non-mammalian vertebrates. So in the first step, I wanted to assess the presence of a full-length copy of PRDM9 in the whole salmonid family. Because as I said, previous studies have identified a full-length PRDM9 in a couple of species, but I wanted to complete this work by using a BLAST approach in order to look for the presence of a full-length PRDM in the whole genome of 12 species representative of the salmonid family. But it appears that salmonids have undergone at least 2 whole genome duplication events in their recent evolutionary history. A first one in the common ancestor of all teleost species about 320 million years ago, and a second one in the common ancestor of salmonids about 90 million years ago. And these multiple WGD events resulted in duplicated chromosomes and multiple paragous genes. And it appears that I did found several paragous copies of PRDM9 in the 12 species that I analyzed. And among these paragous copies, I found exactly one full-length PRDM copy in each of these salmonid species. And this full-length copy corresponds to one paralog in the 4 genera Oncorhynchus, Coregonus, Timalus, and Savelinus, while the Salmo has conserved another paragous copy. So this suggests differential retention of functional paralogs among salmonid species, and the fact that each salmonid has retained exactly one full-length PRDM copy suggests that this PRDM gene is very important in this group. So then I wanted to assess the function of this full-length PRDM copy in the salmonids by reconstructing the Recombination landscapes of 3 species belonging to the 2 groups that has retained a different paragous copy. So in the Coho salmon, Oncorhynchus kisuch, the rainbow trout, Oncorhynchus micis, and in the Atlantic salmon, Salmo salar. And I used a linkage disequilibrium-based method to reconstruct these recombination landscapes. So this method infers the population recombination rate rho, which is proportional between the recombination rate R and the effective population size. And this method offers among the final resolution to detect recombination hotspots by using phased genome-wide resequencing data from a sample of population. And they can infer historical recombination events from over thousands of generations. And so this method relies on the information of linkage disequilibrium between pairs of SNPs. So, for example, when there is a strong linkage between 2 successive variants, this indicates a low recognition, while when there is a weak linkage, it indicates a high recognition. Weak linkage, high recognition, sorry. So from the local information of LD, this population-based method can reconstruct the chromosome-level recognition landscapes. And so here are represented the whole genome recognition landscapes of the 3 species I analyzed. And first we can see that recognition rate vary at different scales through all the chromosomes of the species. When looking at a very fine scale, I found between 17 and 23,000 of recombination hotspots in these 3 species that accumulate about 25% of the recognition, less than 3% of the genome. And so then I looked at the location and the evolutionary dynamic of these recognition events and hotspots in order to elucidate the role of this PRDM9 protein. So first, I looked at the recognition rate at promoter-like regions. So here I represented the recognition rate according to the distance to the nearest promoter-like and open chromatin regions, and we can see that there is no increase of recognition at the location of transcription start sites and TSS that are located in CPG islands. And I compared this level of recognition at promoter-like regions with the CBAS, which has a truncated PRDM protein, and this species shows level of Rho almost 3 times higher in TSS that overlap with CGIs. So the lack of enrichment in promoter regions in the 3 salmonids is consistent with what has been observed in primates, whereas the enrichment of recombination in the promoter-like regions in the CBAS looks like what is observed in species that totally lack the protein. And so then I looked at the evolutionary dynamic of this recombination landscapes by comparing species and populations with different degrees of genetic divergence. And to do that, in fact, I've reconstructed the recombination landscapes of 3 populations of Atlantic salmon. So 1 population in the Gaspésie Peninsula, the GP population, and 2 populations in Norway that are very closely related, the BS population in the Barents Sea and the NS population in the North Sea. And so I retrieved about 5% of shared hotspots between the 2 species of Oncorhynchus, and I didn't detect an enrichment of recognition in the hotspots of the alternative lineage. So here we can see that at the location of the hotspots of Oncorhynchus kisuch, we don't see an increase of recognition rate in Oncorhynchus micis in green. So this means that the hotspots are lineage specific. But is it still true at the intraspecific lineage? So I then compared the 2 distinct geographical lineages of Atlantic salmon, the Canadian and the European lineage. And I found only like 10% of shared hotspots between these two lineage, with again a weak increase of recombination in the hotspots of the alternative lineage. So these two populations might be still too divergent to share their PRDM9 allele. However, the two very closely populations of the European lineage might be close enough to share PRDM alleles and therefore hotspots. And so I retrieved about 25% of hotspots that were shared between these two populations. And to know if this higher proportion was related to a shared PRDM allele, I looked for PRDM binding motifs enriched in the hotspots of these two populations. And so I found one motif, this motif that was significantly enriched in the hotspots of these two populations, and that was also enriched in 20% of their shared hotspots. And this motif was also associated with increased recombination rates, so this suggests that these two populations share a PRDM9 allele. So I therefore retrieved very labile recombination landscapes as compared to the primates that share almost no hotspots, and even the very too closely populations of the European lineage share much less hotspots than the bird species, for example. And finally, project collaborators used direct approaches to directly assess the activity of PRDM9 in the rainbow charts using molecular approaches. And first, they show the presence of 7 PRDM9 alleles based on the polymorphism of the zinc finger domain, which is the domain that determines the DNA binding motifs. And they also showed that different PRDM alleles determine different genome landscapes of double-strand breaks that are associated to PRDM9 signatures. So to conclude, using a combination of phylogenetic, LD-based, and molecular approaches that was realized by colleagues, we confirmed the presence of a full-length PRN protein in the whole family of the salmonid, and that this PRDM9 protein is responsible for hotspot location. And right after my PhD, a recent paper in the corn snake showed that the PRDM protein, which is intact in this group, direct recombination events at PRDM9 binding sites, so that provided a strong insight into the function of PRDM9 in non-marine vertebrates. But now our work showed that the PRDM system was already present in the vertebrate ancestor about 450 million years ago, and so this system is much older than anticipated, and then a further question could it be even older than that? So I would like to thank my supervisors, my collaborators, and I'm also looking for a postdoc, so don't hesitate to contact me, and thank you very much for your attention.
I struggle to remember faces. I struggle to remember names. Sensitive to fragrance. Sensitive to noise. Please communicate directly. And I appreciate these tags so much. It's okay to be me. I never know that it's okay to struggle to remember names and faces, and it's also okay to be women. So I feel very happy to come back to Montreal after 7 years, since I was here at another conference in 2017. And my academic career began in 2016 at Haverford. Then I accidentally learned coding and switched to bioinformatics. Then, because I both have molecular and bioinformatics skills, so I became very employable as a bioinformatician, pet bioinformatician in wet labs. However, during 2020 and 2021, I witnessed too much social injustice in the world, in Philadelphia, in my lab, in my apartment, that I found research no longer fulfilling, so I switched to media, and I worked in animation in China. However, that's when I also realized that I am limited. I have my strength in science. I have a very analytical brain. I even wrote a script for my animation company to process their images, so I know that I can make the most contribution to the world in research. So I went back to science and started my master's in Europe. I actually accidentally, again, got very depressed in Northern Europe because of the winters, but then I got better, and now I'm presenting my project, during which I got better. And constructing high-resolution recombination landscapes from thousands of pollen cells. What is a recombination landscape? I guess you already know, but yeah, recombination describes essentially crossover a recombination landscape is to divide the frequency of meiotic recombination over genomic distance. And genomic distance is also what we call resolution here. And because crossover swaps or recombines alleles along a chromosome, it is a very evolutionarily orchestrated process. And the recombination landscape is a very complicated product of evolution. So to study recombination landscapes, we're able to discover the interplay of evolution and intrachromosomal features and interchromosomal features. So to study recombination and its interplay with intrachromosomal features, we need resolution in order to correlate where recombinations happened and the genomic features, as well as throughput, so that we can generate multiple landscapes for different chromosomes, individuals, or populations. However, traditional methods are lack in both resolution and throughput. So even though cytological methods can enable us to observe crossover in real time under a microscope, we can only observe just a few crossover sites, a few markers, and it's hard to pinpoint where the crossover has happened. And if we use a more higher resolution method, like pedigree method, we have to generate F2 offsprings and grow them, sequence them, and this method also loses the real-time component, so we can only observe zygotes that are viable and survive into viable offsprings. However, our new method will both boost high resolution and high throughput, as well as generate recombination landscapes pre-zygotic. So we're able to discover more than 100 markers per genome and resolving the landscape resolution to about 1 megabase or even higher. And we're able to use a bioinformatic pipeline to apply it to thousands of genomes at one time. How does that? Yeah, and so maybe that's not surprising to you because that's already been achieved in human sperms, but we're working with plants. And the plants' gametes or pollen are difficult to study because of cell walls or seed oil and other chemical stuff, but our lab has already overcome this challenge. So how does this pipeline work? I will give a very quick overview and then go into details. First, we assemble this offspring genome, and then we use parental short reads to phase this genome into 2 diploid haplotypes. We align the 2 haplotypes to each other and call variants, and these are what we use as crossover markers. Then we also generate the single pollen cell reads from the offspring. We align the single pollen reads to one of the haplotypes, and we're able to genotype the individual pollen genomes at the crossover markers. Then we're able to detect crossovers and make calculations, and then we get the recombination landscapes. In detail, that happens this way. We have these 2 genetically divergent parents. We get this heterozygous offspring and the parental Illumina short read. We get a long read from the offspring and then phase this genome using a new technique called trio binning. Then we use a tool developed in our lab called SIRI to call variants between the 2 offspring haplotypes. And then we align the single cell reads to one of our offspring haplotypes. And then we're able to genotype the single cell reads at the marker regions, at the markers. And after that, if we look at one of the genotyped single cell genome and the reference haplotype, we're able to see, oh, there are haplotype switches. Should we call them crossovers? However, reality is always more complicated. So that is what we see, and we can clearly tell we got noise. So what we did instead, and we cannot really call crossover at each of these haplotype switches because we know that in plants only 1 to 3 crossovers happens per chromosome. So we have this statistical model developed by our collaborators to calculate by probability which segment of these multicolored markers are actually one haplotype. So we assign haplotypes to segments instead of to markers. And then we do some more filtering because, for example, this very short segment has only 2 markers. Is that really reliable? And then we also filter out all the segments that are identified by less than 5 markers. In the end, we have this chromosome with a gap, so we can't really say if there is a crossover in the chromosome or not. Then we throw away the chromosome. Eventually, we have these remaining chromosomes, and we can calculate, based on the segment haplotype switches, the number of crossovers, and divide them by the genomic windows. Then we finally get our recombination landscape. So our results are good. Using the trio binding approach, we're able to generate a very high quality genome that is even slightly better than the current reference genome. We have a larger size, less gaps. We have less than 1 mistake per MB per base accuracy, and we have correct scaffolding, we have correct, nearly like perfect phasing, and we have expected genes that are present in our genome, or the buscos, if you heard about it. Then our method is high throughput. So using the single cell sequencing platform, it works by assigning barcodes to individual cells, and sometimes the barcodes capture multiple cells, sometimes multiple, or the barcodes capture something like a trash DNA fragment that is not really a genome. So we also have those like barcode 2 and barcode 3, those are trash barcodes. So we filter them out, but still we have thousands of pollen cells that are real cells, and we can generate recombination landscapes for all of them. And by filtering the pollen cells, we are able to get a high quality cells that have at least more than 100 markers per single pollen genome. And what does this mean for the recombination landscape resolution? Then earlier I mentioned that by counting the number of haplotype switches in the segments and dividing them by the window size, we're able to calculate the recombination rate. However, the reality is still a little bit more complicated. So the segments are not resolved at where crossovers happened. We only can detect that haplotype switches happen between 2 markers. And if we divide a shorter distance, and if we use a window size that is shorter than the marker distance, then we're having trouble to assign the crossover at a definitive location. So what we have to do is to plot the marker distance between all the markers, and what we see is this. And we want a window size that captures nearly all of the marker distances, which is 1 megabase. Then we're able to definitively assign the crossover to this particular window. However, it's still possible to make the marker distance shorter, because sometimes people say 1 MB is like the standard, it's not high enough. So it's still possible to eliminate cells that have very few markers and very long distances between markers, and then we can get an even better resolved landscape. So finally, is all this talking reliable? So we compare. So this is our recombination landscape for Brassica rapa, a very well-studied species. And we're able to see this standard classic U shape of recombination landscapes. And we're also able to see that some of the recombination landscapes do not have a U shape, and they belong to acrocentric chromosomes. And we compared this particular landscape with what is published in a genetic map. And indeed, that chromosome belongs to a chromosome that has an acrocentric, that is acrocentric. And we're also able to see that recombination landscapes with very high recombination rates corresponded with a colored region with a high recombination rate area in that particular chromosome. So our recombination landscapes are correct. And in summary, so our method works by trio binding, by generating an offspring phased genome, and then aligning single pollen read to one of the haplotypes. And by aligning the 2 haplotypes together, we're able to generate the recombination markers. Then, using the recombination markers, we can genotype the single pollen genomes and detect crossovers. Then we do some calculations, and we get a recombination landscape. So, thanks, everyone, and thanks to the people in my group. the people directly who worked on the project, and Raul, who is my direct supervisor, Raul, who is also driving this project with our big boss, Corbinian. And I would also like to thank my support group, my former supervisors and mentors, my therapists and my friends and my pet. They are all very important to my well-being. And a very important lesson I learned from this experience is that I do not always have to cope against setbacks to make progress. I can make just as much progress in love, in a nourished environment. And we just do not have to face everything alone, because everyone is important, and you're not defined by your experiments or failures or what other people say, but by yourself, the days you live, the roads you traveled, your existence, your culture, like everything that comes with you. So if you are struggling, I want to tell you that you are not alone, and thank you very much for listening to my talk.
So good afternoon, everyone. I'm Marion Talbi. I'm a third-year PhD student at the University of Bern, and I'm interested into evolution of recombination rates. Okay, so I assume that if you're here, you don't need to be convinced that recombination is important in evolution, and we already heard like some talk explaining us that, yeah, recombination is variable, and variable at different scale, of course, but the variation I'm interested in is the variation along the genome. So that's the so-called landscape of recombination. And this landscape can differ as well at different scale, right? Between different individuals, but also between different species, or even between different populations. And in this talk, I spoke to you about the work of the first chapter of my PhD that is is online. So I will just present you today a small piece of this work. So if you're more curious, just come talk to me. I'll be super happy to tell you more. The idea is that in this work, we took really advantage of a really, really nice system that this cichlid fish is called Astatotilapia calyptera. And so this fish is encountered in a really tiny crater lake named Lake Masoko that is localized in Tanzania. And so this lake is 50,000 years old, so it's pretty young. And within this lake, there is actually two different ecotypes that start to diverge, as you might see there on the phylogeny. And it's actually an example of ongoing selection. So it's really give like a perfect, framework to study evolution of recombination rates within this context of speciation. So what I start to do is inferring the demographic history of these two different populations. So on the y-axis, you have like the time scale in log, and the x-axis is like the effective population size. And what is very important here, and what I want to bring your attention on, is that they actually shared a really, really long common history. And they actually split only 2.5 thousand generations ago, which is really recently. So if I show you now how those look, the genetic structure of these two different populations, you would have like in square the benthic individual, and in triangle the littoral one. And you see that they cluster differently on the PCA. However, you saw as well, so there is this gray individual that I will call the hybrids one. So there is ongoing gen flow right now. And for the rest of the presentation, I will focus on the blue individual on the left and the yellow individual on the right, because I really want to invest like how does the landscape of recombination diverge between these two different ecotypes. So you might know that, so I'm using LD-based method to infer recombination, and you might know that these methods are not perfect, far from that, and they are really like subject to a lot of bias. And one bias can be, for example, different demographic history, as I showed you earlier, or even like just sampling variance, right? So what we did to try to control for that is first, we applied this subsampling scheme. So from each pool of individual, we subsample in a sample A and B, the littoral population and also the benthic. And I applied this scheme on the empirical data. But what I did is I also used MS Prime to simulate a genetic data under the same demographic scenario that I showed you earlier, for the same evolutionary parameter, let's say like mutation rate, for example. So for each of them, this subsample, I built recombination maps using Pyro. And I will show you one result. So, the y-axis shows the Spearman correlation that is calculated on 2 kb window along the genome. And then in yellow, you will have the comparison between the A and B from the littoral ecotypes. In blue, the comparison between A and B from the benthic ecotypes. And in gray, here, you will have the comparison in the between ecotypes. So let's say sample A from littoral versus B from benthic. And here are the results obtained from the simulation. So from this, we derive like this DMS value that is this difference in the within versus between ecotypes. And we obtain the same value for the empirical data. So then we can really look not only at the absolute value of correlation, but at this relative difference between the simulation that is kind of a control or a neutral expectation and the empirical data or the data I'm working on. So basically what you can see here is that there is a higher difference in the empirical data versus the one of the simulated one. So this suggests that there is divergence in this population recombination rates since the divergence. So the next question was obviously, okay, where do we find these differences on the genomes? And here will be like the recombination distance, and if it's like below the diagonal, is that it's higher between the ecotypes than within, and the opposite below the diagonal. And so this higher divergence between the ecotypes is actually localized on more than 80% of the genome. And I will also speak about this outlier. So we choose them as we assign them as the 5% of the genome with this higher difference in recombination rates. So yeah, now one can ask this question, or at least we ask this question, what drives, or we want to know what drives these differences. And even first, we want to know what does not drive these differences. Because I told you that, so imagine you have like this small fish that arrive in this crater lake, and of course like this littoral morphs start to explore this benthic environment, and eventually they undergo like some divergent selection, right? And selection, due to the effects of link selection, actually erodes diversity. However, I told you that I use LD-based method to infer this recombination map. So I just also want to be sure, right, that this different, we're not just spurious and just do by this difference in the pattern of genetic diversity. So what we did is that we divided the genome into like quantile, so FST quantile and delta pi quantile. So, you know, for example, from 0 to 0.1, value of FST would be like the first point, et cetera, et cetera. And we look at the correlation that I showed you earlier. And what you can see here is that the pattern is really consistent, and even in the value of really low FST or the low delta pi, we actually observe this stronger divergence between the ecotype than within. And what is also interesting is that it's also always like the data obtained from the simulation are always closer from the one than the one obtained empirically. So it's very consistent, and the pattern is consistent. It doesn't seem to be driven by this recent selection that was going on. So looking at the pattern of FST along the genome, we also observe kind of this high area with higher divergence. So, okay, my system doesn't have PRDM now, so I didn't expect to find like rapid evolution of recombination landscape. And I was thinking, okay, maybe something could be okay if we have like some long haplotype segregating in the population since really long time. Maybe since the divergence, they might start to sort, and it could maybe be a reason why, like that rapidly, we would observe some difference in landscape of recombination. So I use a local PCA outlier method to find this long haplotype along the genome. So it would be like region of really low recombination that can be for different reasons, right? Let's say inversion, or maybe like old introgression, for example. And I found like more than 47 blocks localized on more than 20% of the genome, which I think is correct number, is kind of high. And then looking at this outlier that I was speaking to you before, we found actually that this outlier of recombination were more associated into this local PC outlier than what expected and what we expected by chance. So this area actually corresponds to place where, for example, there is a reduced level of recombination in one of the ecotype, let's say, for example, here in the littoral. And if we a bit zoom out, so here you have a haplotype plot. Each line would be an individual, and each row would be a SNP. In red, you will have like the heterozygous site. So you see that we see these really long tracks of heterozygous, and for this block to have an impact on recombination differently between the ecotypes, we would expect that actually there is a higher proportion of heterozygous in one of the types that will impede the recombination to happen. So that's, for example, what we observed here. So we saw like there is more yellow individual with like the heterozygous track. And then something that we want to invest was, okay, is there a link actually between the level of differentiation in this block and the divergence in recombination between the ecotypes? So here, each point is actually one haplotype blocks, and we saw only a weak relation between like FST and recombination divergence. However, we observe a really stronger link with the individual heterozygosity. So what do I mean with that? It's what we expected actually, because so if I come back to this plot, you see like each point on the right will be the proportion of heterozygous site per individual. And for this, I told you to have an impact on recombination, which should observe a differentiation in the proportion of heterozygous individual associated with like the ecotype. And for example, that's what we saw here, and we can derive the p-value from that just using a simple binomial test. And then that's this p-value that we plot against the divergence and the distance, absolute distance in recombination. So to conclude, we found divergence in population recombination rates despite a recent split. We found that recombination divergence was not driven simply by differential patterns of genetic diversity, that it's overrepresented in regions characterized by these long haplotypes, that it's associated with a proportion of heterozygous individual in each ecotype. And so one can ask, okay, could evolving recombination landscape facilitate rapid speciation with gene flow? And that's a question that I will advertise a bit now that we want to to discuss a bit more. So with my supervisor and Diego, a postdoc in Nicolas Salaman's group, we're organizing in Bern at the end of September a workshop on this evolution of recombination landscape, facilitating adaptation. So to discuss a bit the role of evolving landscape in this context. So yeah, if you are interested into that, you can scan that or just come to me as well. And with this, I would just like to thank you for your attention, and my supervisor, Milan and Ole, also our co-author, George Turner, and of course, like the Aquatic Evolution Group and the IE people. And yeah, thanks you. And I'll be happy to answer questions. Yeah.
Okay, everyone, welcome back to our third session on recombination, and we're going to start, I have no clue if this microphone works, it's going to start, okay, with Jasmine Ono, who's going to talk about chromosome divergence and hybrid sterility in yeasts.
Great, thanks. So I put up the University of Nottingham logo to say that I've recently moved to the University of Nottingham to start my own group. Look me up there now. But this work is all work that I did during my postdoc with Duncan Gregg at UCL. And when I say I did it, I really mean that Dave and Ozan did most of it. So really, they're responsible for lots of this. I was waiting for that bell. Okay. So some of the big questions that I'm interested in are, why do species exist, and what maintains species as separate groups, and how does this evolve? And I promise this will get around to recombination. So I study speciation using Saccharomyces yeast, so budding yeasts. And right now I'm going to talk to you guys about some experiments where we compare Saccharomyces cerevisiae, so like the main yeast that most people think about that makes beer and wine and everything, to its closest relative, Saccharomyces paradoxus. And the main species barrier between these two species is hybrid sterility. So you can make a cross between them, an F1 cross that's heterozygous. It's totally happy to grow mitotically, but as soon as you try to put it through meiosis, that doesn't go so well. So you get less than 1% gamete or spore viability. And I might use the words gamete or spore interchangeably, warning now. Okay, so we were interested in investigating the potential causes of this hybrid sterility. And the first one I'll talk about is chromosomal rearrangements, just because this doesn't seem to be a driving force in Saccharomyces speciation. So there's no large rearrangements or differences in ploidy that are fixed between species of Saccharomyces, despite the fact that we see a lot of variation within species. So it's obviously important for some types of evolution, but not for speciation in this case. Okay, so the second part that I'm going to focus on is chromosome missegregation during meiosis. And I should say that before we did this, we already kind of knew that this was going to be a major factor in Saccharomyces hybrid sterility. And I'm going to do a little review of meiosis, even though you're in a recombination section and maybe you know this, but it's really good to all be on the same page because it's going to get kind of specific. So what I'm representing here is what happens normally during meiosis. So I've put a diploid yeast. Oh yeah, I forgot to say that this is my cartoon representation of yeast, the thing with the little bud. So on the left-hand side is a diploid yeast with only one chromosome represented. And I've colored them in two different shades of blue, so the darker blue and the lighter blue, to say that they're not so different from each other, these two different copies. So what happens first during meiosis is that these chromosomes are replicated, and then we get the crossing over and recombination. I'm going to zoom in on that because I realize that probably you can't really see my little cross that I put between these homologous chromosomes. And so a lot of what we were talking about in this session is how recombination generates more variation, and it has been also mentioned, but in case you don't know, another really important role of this crossing over and recombination is that it actually creates physical tension between the chromosomes as well. And this is really important for the segregation process at this first meiotic division. So this helps to ensure that these two different, the two homologous chromosomes go to opposite ends of the cell, and then they separate at the first meiotic division, then the two sister chromatids separate at the second division. You get one copy of the chromosome per gamete. Great. So the problem happens in hybrids where now our chromosomes are quite diverged from each other, so that's why they're in blue and red. We no longer get that crossing over at the first meiotic division. So now there's opportunity for these chromosomes to kind of go wherever. And so some of the time, they'll both go to the same pool and end up in the same cell after this first meiotic division. Then they segregate normally at the second meiotic division, and we end up with two spores with two copies of the chromosome and two with none. And the ones with none are dead because every chromosome in Saccharomyces is essential. And what I represented there was what happens with just one chromosome. But you can kind of see how the problem gets worse as we add in more chromosomes. So for example, with two chromosomes, a straight and a squiggly one, what I've represented here is normal segregation where it all worked out well. But instead we might have missegregation, and they could actually end up in different cells if they've gone to different poles at that first division. And now all of these are dead because they're all missing one of their chromosomes. Yeast actually has 16 chromosomes, and so if we kind of do the math, if things are segregating randomly during meiosis, this could actually explain pretty much all of the hybrid sterility. Okay, so I mentioned that it was known before that chromosome missegregation is important in yeast hybrid sterility, but the methods that were used previously to determine this weren't very quantitative because they depended on growing up the things that were viable. And so first of all, you're not measuring those things that aren't viable. Second of all, as you grow them up, you actually might lose these aneuploid chromosomes, so you're not getting an accurate count. And so what we've done is taken advantage of a new technology where you're able to express fluorescent proteins in those spores themselves. And I should have said, sorry, so this cute little, I think it's cute, little picture on the right is tetrad. So in yeast, those four products of meiosis get packaged together in a sac very conveniently, so you can actually look at the segregation directly in that tetrad. Okay, so we've marked the dark blue in green and the light blue in red. And then when we look under the microscope, we should see two green cells and two red cells if all of the segregation worked out well. And here's some real pictures. So on the left, we see that we do have two green cells in each of our tetrads, yay. We have 2 red cells in each of them, in the middle, great. And on the right is when we merge those 2 photos. You see most of the time, those green and those red cells are different from each other. Sorry, the green and the red labeled chromosomes are going to different cells, but sometimes they don't, and those appear as yellow in that merged photo. And so what we did is we did this same process in quite a few different crosses and just counted up how often we saw each type of segregation. Oops. Oh yeah, sorry, I was supposed to say, you got that, yellow. Okay, so all of that data is represented here, and they're shown by the black points. So the black points show the non-destruction rate, and I should have said that this specific type of missegregation I was telling you about is also referred to as non-destruction. And with increasing, so we're showing here the black points with the non-destruction rate, with increasing genetic distance as you go along the x-axis, and in the red is just like a calculated estimated viability based on that non-destruction rate. And so what we see is that for our between species crosses on the right, we actually have really high non-destruction rates, so almost 0.4, and with 0.5 being completely random. And so we find that the observed spore viability in our interspecific hybrids, that's about 1%, actually can be mostly explained by this non-destruction. And I did also just want to point out that these points on the left-hand side are within species crosses of Saccharomyces, and we see the same pattern even at lower levels of divergence. So what seems to be happening is that this anti-recombination mechanism is preventing crossing over between the diverged sequences. So what happens is there's a double strand break and you get the strand invasion, but there's mismatches between your different strands of DNA. So that's represented by a purple star. You then have proteins that come in and identify those mismatches, this MSH complex, and then that recruits other proteins to say, no, this isn't good. This might be something totally different that we should not be crossing over with. Let's get rid of that. So to summarize what I've kind of told you about so far, sequence divergence is associated with sterility of hybrid yeast, where we see that the anti-recombination system inhibits crossovers between diverged sequences. The chromosomes then missegregate during meiosis, and because of this, there's missing chromosomes in some of the spores, and this leads to sterility. And I also put a note to remind myself to tell you here that we do have really high sequence divergence between species of Saccharomyces. So it's about 13%. So that might be much higher than what you're used to thinking about. Okay, so chromosome missegregation during meiosis is definitely playing a role, and this is due to the genome-wide sequence divergence and explains most of the hybrid sterility, but not quite all of it. And we were really interested in also looking at genetic incompatibilities. But to be able to, so genetic incompatibilities is this kind of like idea of speciation genes that a lot of speciation biologists think is like the main genetic model for what keeps species separated. So the idea is that you have separated populations, they get different mutations, so small a in the red one, small b in the blue one, and when these come together, these new combinations of alleles come together in a hybrid, so small a, small b in the purple one, sometimes they might result in low viability or low fertility or sterility, and then we would refer to these as speciation genes. So we wanted to map these speciation genes, but we have a problem because we have like pretty much no viable spores. And so what we did is we targeted these 2 parts of the anti-recombination system, and we repressed them specifically during meiosis. And so, part of the text is missing. Anyways. So the result of that is being shown here. So on the y-axis, we get our hybrid spore viability, and along the x-axis, we have our different strains. So the wild-type hybrid is all the way on the left. It has like no spore viability. In the middle are the 2 single mutants, and then on the right is the double mutant, and we see that we can get up to like almost 33% spore viability. And of note is that this is actually higher than for some within species crosses. So now that we're able to get viable spores out, we do another yeast genetics trick where we actually take those spores out of the tetrad and put them in specific places on a plate so that we can recover them after they grow up. So that's what's being shown on the right is the plate after those spores have grown up, if everything actually grows pretty much. And so you can see there's 4 colonies as you look down. Those will be the 4 spores from a single meiotic event. And then what we did was we only chose to use tetrads that had 4 viable spores, because that implies that all of the chromosomes had to have segregated properly during meiosis, and now we have nice euploid hybrids that we can actually use for mapping. And so we've sequenced a bunch of tetrads, and this is like a representation of the sequencing data where the chromosomes are along the y-axis, and for each chromosome there'll be 4 lines, each of which is one of those 4 spores, with the 2 genomes colored in red and blue, and that's just to show like we do see some amount of recombination, and it breaks the genome up into some smaller regions, and then totally skipping over how I did this, I took those regions and I tested them for statistical underrepresentation of hybrid types compared to parental types to try and map incompatibilities. Oh yeah, these incompatibilities. So then the results of that are shown here. Sorry, it's a bit light, but here the chromosomes are shown all around the circle, and a line is drawn between regions that had significant underrepresentation of hybrid types. So that's where we think there's an incompatibility. And so we find that there are probably 4 incompatibilities that we were able to broadly map using this new technique. So we find that chromosome segregation is the main cause of hybrid sterility, but genetic incompatibilities can potentially explain the rest. And thank you.
Okay, so I'd like to thank Kieran and Susan for including me in this session. It allows me to talk about something that I usually talk about at poster sessions or over beer or whatever, and actually now it forced me to formalize it into a 15-minute talk. So hopefully what I say makes some sense, and if I'm wrong on things, please tell me. So I'm just gonna start with that as a caveat. So as we know, regions of reduced recombination are very important, and we're all excited about them because they can hold together linked loci, multiple alleles at linked loci that are adaptive, and prevent recombination between them, forming these super genes that do all kinds of different things across the wide swath of life. So that's great and all, and suppressed recombination is fun, but there are some issues with it, in that when you have suppressed recombination, this can limit the ability of loci to evolve under selection through interference between different loci through the Hill-Robertson effect. And relatedly, you can have negative effects due to things like Muller's ratchet, where if you have a region of suppressed recombination, you're going to build up deleterious mutations over time. They can't be easily purged by recombining and getting rid of them. So that's the downside of it. But the thing is that different types of suppressed recombination in the genome operate in different ways, and so these effects are going to be different. And so what I want to do today is contrast 2 major types of regions of genomes that can suppress recombination, but I want to argue that they're very different in how they work. And this may be obvious to a lot of you, but I've definitely seen a number of talks where this was not obvious, and so I just want to talk about it here today. So what I'm going to do is contrast chromosomal inversions, which we just have heard a lot about, versus pericentromeric regions, which we haven't talked too much about today. And pericentromeric regions are the regions in the center of chromosomes. And so for a lot of the organisms you work on, they may be big and they suppress recombination. And in things like the plants that we work on, they also include a lot of genes in them too. So they universally suppress recombination, sometimes include up to 30% of the genes in the genome. And so they're very different from chromosomal inversions, and I want to tell you how. And I also want to tell you how just doing the empirical work in my lab has kind of brought me to this conclusion. So before I go on, I just want to state this hypothesis about the differences between chromosomal inversions and pericentromeric regions. And you can think about pericentromeric regions or other regions of suppressed recombination that are not structural variants. And the reason why I think that chromosomal inversions are so often associated with supergenes or are involved in local adaptation is because they hold together haplotypes of adaptive alleles while minimizing the interference of linked alleles under selection, as well as the accumulation of deleterious alleles. And so I'm going to try to tell you why that's the case. And again, this may be obvious to a lot of you. So where these ideas come from is from pondering results that I've gotten from multiple plant systems I've worked on over my career, including monkey flowers, panicum grasses, and data sets coming from Arabidopsis, because I don't like to touch Arabidopsis, I just work with its data. So the system that I've done the most work on thinking about this is in the coastal and inland ecotypes of the yellow monkey flower Mimulus cutatus, which is adapted to different habitats in California, where its inland habitats dry out in the summertime. These plants are annuals, they have to escape from drought, whereas ones that are near the coast can actually live for multiple seasons as perennials because they have lots of fog, they have lower temperatures, and so they've evolved these locally adapted differences. About 15 years ago, back when I was in graduate school, I discovered that there was a large chromosomal inversion that underlies a lot of this divergence between these annual and perennial populations. All of the perennial ones in blue there along the coast have one orientation of the inversion. All of the annual ones have a different orientation of the inversion. And that's fine and cool to like see that pattern. But we actually did reciprocal transplant experiments and tested the effect of this inversion in the field, and it has a big impact on adaptation to both of those habitats. It leads to flowering time escape from drought in inland habitats and multi-season survival in coastal habitats. And so it's important for local adaptation to those different environments. Now, for years, I've been trying to track down the breakpoints of this thing, but finally with genome sequencing technologies getting to where it's at, we've been able to actually map this inversion using nanopore sequencing. So Leslie Kohler, a postdoc in the lab, has done this work. And what's really exciting about this is that not only did we map this chromosomal inversion, but we discovered a new inversion within that inversion. It's about 250 kb, which I'm going to tell you a little bit more about. So in order to understand how these things are differentiated and to look for like the loci within the inversion that might be actually underlying its phenotypic effects, we conducted whole genome pooled sequencing where we pulled lots of coastal populations, lots of inland populations together and sequenced their genomes and then looked for allele frequency outliers. And so if we look into this region where the inversion is, so there's this orange region with this green line on it. That orange region is where this large adaptive inversion is. The green region is where this new nested inversion is. If we look at divergence there, we notice that the nested inversion actually has a lot more divergence than the rest of the inversion or the genome-wide average, which is down below. So it looks like it's ancient and it may have been a supergene itself that was then later subsumed by this larger inversion, which is now one of the most important loci in this system for differentiating different ecotypes. In addition, by using this allele frequency and outlier data, we were able to localize potential candidate genes, including this key gene, GA20 oxidase 2, which is in the gibberellin hormone pathway in plants. So you can think of this as just growth hormone for plants if you're not a plant person. And we were excited about this as an outlier, looked at it in more close detail. And what was really interesting about it was that there's not only did it look like it is differentiated between these populations, but it looks like it's possibly gone through a recent selective sweep in the coastal populations. So it has a much, the Tajima's D statistic is in line with that. It has much lower diversity in those coastal populations. So it looks like it's possibly been under recent selection. Now we've taken this gene and we've been working with it because we know about all these supergenes, but we often don't know how these supergenes work. So using the awesome power of the monkey flower, we can now do transgenics and like transform these plants simply by injecting the ovary with agrobacterium. So we skip all of the problematic things of tissue culture regrowth. And so we can now engineer things like our coastal, perennial, and inland annual monkey flower. And with that, we were able to both knock out with CRISPR and overexpress this GA20 oxidase 2 gene. And what it does is actually somewhat converts the plant from a coastal plant, which is the wild type, to an inland type plant just by overexpressing it. We now have the knockouts, which we're going to do promoter swapping with to see if it actually was a gene that changed that affects the chromosomal inversion. Now, the thing that I've been pondering about with this is this idea that maybe or maybe not this gene within the inversion has been under recent selection. So you're wondering, I wonder to myself, how can this be the case if this is a region of suppressed recombination? And so what I want to lay out is just a very simple model of thinking about how these inversions actually evolve, which is to think about that the coastal habitat is mostly homozygous for one orientation of the inversion, Inland habitats are mostly homozygous for the other orientation and inversion. And then we have occasional hybrid zones where we see populations that have both inversion types in them. Now, if we think about where recombination is actually suppressed, it's only actually suppressed in those heterozygous individuals in the hybrid zones. And so in places like coastal habitat or the inland habitat, we have free recombination occurring within that inverted region. We can have selection on individual loci. They're independent of each other. And so we can actually have the accumulation of more adaptive alleles easily without having Hill-Robertson effects. And also we shouldn't expect to get a lot of deleterious alleles building up in those inversions. So they have this great benefit of being able to hold together adaptive combinations, but also to purge alleles that are deleterious. Now, I want to contrast this with paracentromeric regions. So these regions of the genome are something that I stumbled across through working on these panicum grasses when I was a postdoc in Texas, and I've worked on since then. And these things were really surprising to me at first, that you have these huge regions of the genome that just don't recombine. It's not that they're inverted or anything like that. They just don't have recombination in them. They're filled with a lot of repeat elements as well as centromeric repeats. So if you look here, we have like the 9 chromosomes of panicum holii here. And you can look down at what makes up these different parts of the chromosomes. And so the genic regions are primarily in the arms of the chromosomes, but there still are genes in the middle of in these paracentromeric regions as well. And it doesn't matter where this paracentromeric region occurs. So if you look even on chromosome 7, where that paracentromeric region is at the end of the chromosome, that still suppresses recombination heavily there. So these regions just simply suppress recombination in a big way, and they're going to be fundamentally different from chromosomal inversions. Not only do they have genes in them, but we also map loci to these paracentromeric regions. So we map traits to them. So in this case, Acer Van Wallendale, who's here in the audience, did a lot of great work mapping loci that are involved in rust resistance in switchgrass across different field sites throughout the center of the United States. And if you see that big peak on there, on chromosome, on that 3rd chromosome over there, we see repeatedly that is associated with rust resistance, and it's associated in multiple different field sites, and that is a paracentromeric region. So something in there is causing this effect. So, and people have seen this in other systems as well, where you have paracentromeric regions affecting traits, and so they do have importance as well and shouldn't be ignored. Now, if we go back thinking about that model that we had before and thinking about where we have recombination occurring, before we had it only one place suppressed recombination, but in this case with paracentromeric regions, it's suppressed everywhere. So it's fundamentally different than thinking about chromosomal inversions. So here's once again a reminder of like what is going on with the inverted loci, where we only have suppressed recombination in that hybrid zone area. And again, inversions have a great benefit, especially in these local adaptation contexts, in that they can hold together linked alleles, but minimize the interference of linked alleles under selection and the accumulation of deleterious alleles. So I go and I tell this to various people when I talk to them, just because it's a fun thing to talk about. And one time I was meeting up with Steve Schaefer at Penn State University, and he's like, that's a nice idea and all, but you really need to model it. And he took me over to a binder where he had printed out the full manual for SLIM, and he had me flipping through it. And I was like, okay, I am going to do this, and I'm going to work on this. The next month, the pandemic started. I was at home with my kids for 400 days straight, and I haven't really gotten back to this. But anyhow, I just like to say that in conclusion, paracentromeric regions and chromosomal inversions both suppress recombination, but they have fundamentally different dynamics. Context-dependent recombination suppression is what really matters, and that's important. And adaptive inversion complexes can build up over time, so as well as we saw with the other supergene example with the ants. I implore you, if you're a theoretician, please model this. I haven't been able to find a model about it. I've been looking, or send me the model that exists, but it would be very useful for me. So thank you very much, and I'd like to thank all these people who have been helpful to me over the years.
Quick questions? Yes.
fit into this and thinking about the different types of translocations?
Yeah, so translocations are, it's a different thing. They'll suppress recombination and they'll tie stuff up completely, but they're often underdominant in their effect, and so they have a different dynamic. And so I'm thinking about situations where it's like if you bring those different types together, you're not having that big negative impact. Yeah.
I wondered if all plants' chromosomes are like have the centromere in the middle, because the data you showed with that surface type plot, there's like a pattern of lower recombination in the middle of chromosomes in general. Do you have any acrocentric plants you can like test that with?
Well, there's the one where it's the paracentromeric regions at the end, and it suppresses recombination as well. So it's not just being at the center; it's actually those type of regions suppress it. Yeah.
Okay.
Do you have any sense of like how often inversions are forming? Because obviously, like getting a good inversion depends on getting an inversion at all, and they have to be in the right place. Whereas everything is paracentromers.
How often inversions form, I think it varies greatly across different clades of organisms, but yeah, I'd be happy to talk to you more about that. All right, let's get to the next
Thank you.
Hi everyone, thanks so much for coming to my talk, and thanks so much to the organizers for putting on a really great symposium on recombination. Today I want to tell you a bit about how aspects of host biology, and in particular ecology, give rise to variation in the frequency at which pathogens recombine. I'll also tell you a bit about the consequences of this variation for disease emergence. And today I hope to convince you that pathogen recombination is somewhat special and that it provides a particularly useful context in which to understand variation in recombination, the causes, the consequences, and its evolution. One reason for this that I think we're all familiar with is the fact that recombination gives rise to differences in evolutionary potential, and in the case of pathogens, those differences are of public health concern. This is clear if we think back to 2009 with the emergence of the swine flu, H1N1. It turns out in this case, a particular kind of recombination that occurs in segmented RNA viruses called reassortment was really pivotal in this process. And that's actually the weak form of the statement, because reassortment was key in the emergence of every influenza pandemic after 1918. And flu is not the only virus to have a highly, highly retriculate recombinant evolutionary history. SARS-CoV-2 and the SARS-like coronaviruses from which it descends are also really highly retriculate in their evolutionary past. Here, I'm showing you an ancestral recombination graph that was inferred from the present-day sequence data for these SARS-like coronaviruses, and every horizontal line is a recombination event. It's really, horizontal lines all the way down. And some have actually estimated that there were about 2 to 5 recombinations per synonymous substitution in this family. And that's really, really, really shocking when you consider that the evolutionary rate is 10 to the -3 synonymous substitutions per site per year. The other reason that it's somewhat shocking is that the process of pathogen recombination is very constrained. In particular, you need to get genotypes that co-infect the same host in order to recombine. The way that this works in RNA viruses is even stronger because you need the genotypes to co-infect the same host cell. And at that point, the way recombination works is you've got this RNA polymerase that's moving along one of the templates, it disassociates, it binds the other template, and through this template switching process, you get the production of virions with mixed ancestry. A similar thing happens with reassortment in these segmented RNA viruses, but you swap whole gene segments. Again, mixed ancestry results. And in both cases, I want to emphasize that co-infection is not just co-occurrence of genotypes in space and time. It is stronger than that. The latter condition is necessary for any kind of recombination in any system. But the reason co-infection is different is that a host is not just a patch. It is a patch that experiences ecology and population dynamics, and these feedbacks between the host ecology and, in particular, pathogen recombination during co-infection are really salient. And this sets the stage for the question that I'm going to try and answer in this talk. In what host taxa do we expect recombination between genotypes of a focal pathogen to be most frequent? And I think this question is evolutionarily interesting, but I also think it has some public health concern because there's quite a bit of attention that's being placed in this evolutionary emergence world on if there are hosts, some kinds of hosts, that are more dangerous than others, that are more likely to cause future pandemics. And the answer to this question is, based on the available data, yes, there are host ecological profiles that correlate with spillover and emergence risk. And that is the kind of context in which we're asking this question about where the recombinants are and if that recombination is important for emergence. And I'm going to answer this question using kind of a twofold approach. First, I'm going to build a mathematical model of the process of viral recombination that makes explicit a lot of these eco-evolutionary feedbacks. And then I actually was fortunate to get my hands on some avian influenza sequence data against which I could test the predictions of the theory. So I'll tell you a bit about the model. This is super at a distance, so love to chat more if anyone has questions. We imagine that there's a closed host population. Here I'm showing all the susceptible individuals. It's well mixed. Individuals can acquire, and I should mention that the hosts are not evolving, but they can acquire infection with one of 4 pathogen genotypes, and these pathogens are evolving at these 2 loci, which are involved in transmission. Mutations can also arise. So here, the 0,0 genotype gives rise to a 0,1 genotype. Recombination can also occur, so that, say, a 0,1 and a 1,0 find each other during co-infection, they recombine, and you get the production of a 1,1. You could have also gotten the production of a 0,0, or yeah, a 0,0. But crucially, for both recombination and mutation, we assume that you get the wholesale conversion of the host to either the mutant or recombinant type. With respect to fitness, we make the assumption of local adaptation. If you carry a 1 as a pathogen, that makes you better at transmitting in humans this canonical future host, but worse at transmitting in the host reservoir. The opposite is true for zeros. That makes you good at transmitting in the kind of historical host, but worse at getting into humans and certainly between them. As a result, 1-1s are best at getting to humans and between humans, 0-0s, the complete opposite. They're not very good at that. The kind of last assumption I want to draw your attention to is the fact that genotypes in this model are fully cross-reactive. What this means is that immunity is kind of stupidly simple. The minute that a host in this reservoir population sees one of the genotypes and has immunity, it has seen them all, and until that immunity fades, is immune to them all. And the model goes on to make a pretty surprising insight, gives rise to a very surprising prediction, which is that recombination is most frequent when hosts are short-lived and infections are acute. And it's surprising, I think, because at the individual level, the opposite is true. If I'm a long-lived, chronically infected bat with one genotype, the opportunity to acquire a second genotype and for those two to then recombine is greater than if I was shorter-lived and more acutely infected. This is really a reflection of the fact that the individual level logic doesn't scale up to the population level. What's happening at the population level is a bit different. Insofar as the density of co-infections, what matters for recombination, is proportional to the density of infecteds. That's what our model tells us. And the density of infecteds is inversely proportional to the mean lifetime of a host. This second result is not even mine. It is well known in the evolutionary epidemiology world that that is the case. And the logic for that second proportionality is that shorter lives, or equivalently more turnover at the population level, give rise to more susceptibles, and those susceptibles are fuel for future infection, and those infections give rise to more co-infections, and as a result, more recombination. A kind of second thing pops out of this model, which is a consequence of the fact that recombination is strong at one end of the life history extreme. It turns out that because of this, linkage disequilibrium, the associations between these 1 mutations, is greatest at the other end of the life history extreme. I'll mention that LD depends on a few other things, namely epistasis in yellow and the variance in allelic state in green. Blue is the effective total rate of recombination. But no matter, it turns out when you work out the full kind of expression, you get this really nice prediction for D, which is that it's increasing as a function of the mean lifetime of the host, at least in absolute value. And I went ahead and I tested this prediction using avian flu as a system. You might have heard that H5N1 recently made its way into cattle for the first time. It's really scary. There's some really cool evolution happening in the background. And I'm really just interested in flu because it turns out to make a really good system in which to test this prediction, because the test requires quite a few things be satisfied. Namely, we need multiple hosts. We need hosts that differ in their mean lifetime. We need a pathogen that is recombining or reassorting at least some of the time. Flu is segmented, so it reassorts. And the big thing that you need is a lack of population structure. And that's just because you need to compute linkage disequilibrium and then make comparisons of linkage across these different host taxa. And flu fits the bill for a really interesting reason, which is that the wild birds it normally infects are flying up and down, at least in North America, these main flyways And within a flyway, there's very little population structure. And between flyways, there's quite a bit. Previous modeling has actually gone on and shown that what really matters for structure is whether or not a sequence is found east or west of the Rocky Mountains. The Pacific flyway is a population, and all of the other flyways are a population of their own. And so with this in mind, I retrieved all the H5N1 sequences from GISA from the last couple of years in all avian hosts. I did some simulations to determine cutoffs for all the downstream bioinformatic analyses, because the LD comparisons, it turns out LD has somewhat poor finite sample properties, so you need to be a little bit careful. All the analysis I'm going to show you is with some pretty conservative cutoffs. I removed a bunch of sequences that would be problematic to analyze because they say didn't have information about the host species. And then I looked at all of the hosts in both of these, two populations, east and west of the Rockies. And in each host, there are 8 segments of the flu genome and a bunch of sequences associated to each segment. And for each of those segments in each of those populations and each of those hosts, I aligned the sequences to each other. And then I looked at each alignment, computed some minor allele frequencies, and crucially, linkage disequilibrium between the mutations that are found on different segments. And remember, this is a virus that reassorts, so that's how we're going to get these differences in LD in addition to epistasis. And just as a reminder, this was our prediction. LD in absolute value is an increasing function of mean lifetime of the host. And here I'm going to actually use body size as a proxy because mean lifetime data are unfortunately not available for a number of these species. And there are many y-axes that I could have used, from just regular raw LD to D normalized by the maximum value it can take given the allele frequencies. It turns out, across the board, the sequence data are generally consistent with the predictions of the theory. I'm just going to show you the data for D prime. And the data show pretty clearly that mutations do become more and more linked as you move from the short-lived hosts like ducks and chickens, which are smallest as well, to the long-lived hosts like bald eagles in this case. And I think one really compelling aspect of this data is that the ducks, which I think are in red, have somewhat low kind of residuals relative to the regression. And one reason for this might be that ducks host a number of other avian influenzas, not just H5N1, and there has been previous evidence of reassortment between subtypes in addition to reassortment within a subtype, as we have measured here. And with that, I just want to acknowledge my advisors, Matt Osman and Nicole Medeo, both of whom are just awesome. The Osman and Medeo labs, also amazing. And I really want to also acknowledge Shishi Yang and Elsa Mikkelsen for some really stimulating and useful discussions about the bioinformatic analysis. I also want to thank you for coming to my talk, and I'd be happy to answer any questions.
Questions? Yes?
If I understood, you were modeling recombination probabilities of viruses within one virus population?
Yeah.
I think the pandemics result from recombination that's more like horizontal gene transfer between two. Could you talk about whether those results will apply?
Yeah, absolutely. I think that that's a really good point that in recent memory, most of the, at least for flu, most of the reassortments that have triggered pandemics have been because these different antigenic subtypes have come together. There's also good evidence that the reassortment between subtypes is, at least at a broad scale, correlated with reassortment within subtypes. So I think it's still a pretty useful metric for evaluating risk. Any other questions? Shoot.
Thanks, I love the talk. So you're finding that acute infections have more recombination seems counterintuitive. Do you have any insights on what this says about the conventional wisdom with SARS-2? There are variants of concern you think come from the chronic ones?
I think that's an excellent question. I am also somewhat perplexed by the fact that acute infections give rise to more infections at equilibrium. This is actually also a result in the evolutionary epidemiology literature. And in this model, I think it has to do with a kind of weird phenomenon where because of the buildup of recovereds through the acuteness of infection, you can actually free more susceptibles. I think there are some feedbacks, and I'd love to tell you a bit about them later.
Any more questions?
Oh,
Okay.
thank you so much.
Thank you very much.
Okay, cool. Thanks everyone for sticking it out to the end of the day, and thank you to the organizers of this very interesting symposium. So, sexual reproduction is a nearly universal feature of eukaryotes that we see across many different parts of the eukaryotic tree. But despite this fact, there's a tremendous amount of diversity in the modes of sexual reproduction across taxa. So we have hermaphroditic species, we have dioecious species, and we have multiple different forms of sex determination across these groups. And today, I'm going to be focusing on the two most common forms of chromosomal sex determination, which are the XY and ZW systems that we're all familiar with. And one thing that you'll notice when you look across these different taxa is that there are turnovers between these different systems. So you see shifts between different XY systems, between different ZW systems, and then also between those two. And there's many examples of this. This is just a recent one that I like. In willows, where there have been multiple origins of ZW sex chromosomes from an XY ancestor. So why does this happen? Why are sex chromosomes turning over? Before I get into the details of that, I'm just going to prime you with the classic model of sex chromosome evolution, which I'll go through quickly, as many of you are probably familiar with it already. But basically, you have some sex-determining factor arise on a pair of autosomes, and then once that happens, it becomes beneficial to link sexually antagonistic variation to that locus. So if you have a male determining factor, you want to sort of attach male beneficial mutations to it. And that selects for recombination suppression on that haplotype, which eventually leads to differentiated sex chromosomes that degenerate over time. Okay, so given that this is happening, there's a few different models of sex chromosome turnover that have been proposed. I'm considering all of these in this project, but for the sake of time today, I'm going to be focusing mostly on the hot potato model. So the way that this model works is you start with your ancestral sex chromosome pair. In this case, it's an XY system. The Y chromosomes degenerating over time, accumulating deleterious mutations. And then you also have your autosome, and then a dominant sex determining factor arises on this autosome. And if this autosomal haplotype is less loaded than the Y chromosome, then you can move your sex determining apparatus over to that autosome and get rid of this loaded Y chromosome. So the Y chromosome is the hot potato in this model. There are some limitations to this model. So it predicts that shifts in heterogamety, so from XY to ZW, should be selected against. And it can generate endless cycles of transitions, but we don't always see these in the real data. And I don't want to just pick on hot potato. These models in general have a hard time explaining the dramatic variation in rates of turnover that we see across different groups. So I'll just highlight a couple of examples to really hammer that home. So in this study on the genome of the German cockroach, they found that the X chromosome of the German cockroach is homologous to the X chromosome of Diptera, despite 400 million years of evolutionary divergence. So that's pretty dramatic. And then on the other end of the spectrum, we have the situation in cichlid fishes. And in this figure, I'm showing you across a bunch of different taxa, all the sort of chromosomes are lined up. And each triangle is a ZW system, and each square is an XY system. So you can see that there's just a ton of variation in what the system is and what chromosome it's on. Okay, so why is this happening? I'm going to propose that one important factor might be the genomic landscape of recombination, because we know that this is something that's highly variable across taxa. This is just a sampling of recombination landscapes estimated in different flowering plant chromosomes. And you can see that there's quite a lot of variation here. And I'm especially interested in this phenomenon because this landscape can often differ between the sexes. So this is from a meta-analysis showing that the male landscape of recombination is often more biased towards the telomeric ends of the chromosomes than it is in females. Okay, so how might this interact with the hot potato model of turnover, for example? Well, let's say you have a poorly drawn dip in local recombination rate in the middle of your autosome. Maybe your sex modifier gets lucky and arises in a relatively unloaded part of this chromosome. It can sort of spread for longer, at least initially, on this sort of lucky haplotype. Alternatively, it can arise on a haplotype that has a lot of mutations, and it'll be harder for it to recombine off of that haplotype because it's lower recombination, and that might actually inhibit turnover in this case. Okay, so the question I'm asking in this study is, how does the landscape of recombination affect the probability of sex chromosome turnover? And I'm going to investigate this using the simulation software SLIM. And just to give some context to David's talk, the SLIM manual is like 1,000 pages long, but it's a great read, so I would highly recommend it. Okay, so here's the general simulation approach, and this is pretty simple to explain, but actually kind of complicated to set up. I start with a pair of non-recombining X and Y chromosomes and a pair of autosomes. And I sort of just let them do their thing for a while and let mutations accumulate, in this case, deleterious mutations. And then after 8N generations, I'm going to introduce a dominant sex-modifying mutation. So this is a mutation that basically overrides the existing XY system in whatever inherits it. In this example, it's a feminizing mutation, so this would be an XY to ZW turnover. And I do this at an initial frequency of n over 5. I have to do this for computational performance reasons. If I did 1 over 2n, I'd have to do like 1 million simulation replicates, and that would take the rest of my life. So we can't do that. So then I let this keep going, and then over time, the dominant sex modifier is either fixed or lost from the population. And then I just record how often each of those things happens at 16n generations and repeat this many, many times. So I'm simulating 4 kinds of turnover. So there's heterogametic turnovers, which are XY to ZW, and non-heterogametic turnovers, which preserve the XY system. And then there's homologous turnovers, which stay on the same chromosome, and non-homologous turnovers, where it moves to a different chromosome. So first I'm going to show you results for the non-heterogametic turnovers. Okay, so I have this figure on the x-axis. I have a few different population size conditions. And on the y-axis, I have the fixation probability of this mutation that I'm dropping in at 8n. So I have some control mutations. These are the green and purple lines that are just like normal mutations that don't do anything. The green line is that mutation dropped on an autosome. The initial frequency is 10%. It has a 10% fixation probability. Population genetics works. That's all good. The purple one is the same thing, but on the X chromosome. This is a bit higher because the X chromosome has a lower population size, basically, but I'm introducing it to the same number of individuals. So it has a higher initial frequency. Okay, and then these red and blue lines are the masculinizing mutation, the dominant masculinizing mutation, dropped on each of those 2 chromosomes. Okay, so they're above the neutral expectations, and that fixation probability increases with population size. So this is consistent with the hot potato model, right? There's selection to get rid of that loaded Y chromosome haplotype, and that favors the new dominant sex modifier. So that's good. I can recapitulate the previous modeling work that's been done on this. Okay, and then for heterogametic turnovers, the green and purple lines are in the same place, but now the blue and red lines have shifted down, and they're like kind of below the neutral lines. It's actually a bit more complicated because the neutral expectation for those mutations is not the same as the neutral expectation for a normal mutation. But anyways, the main takeaway here is that these have shifted down, and this is consistent with selection against these unfit sort of YY genotypes that you need to produce to get an XY to ZW transition. Okay, so that's also consistent with the model. So that's good. Okay, I'm moving quite fast here, but that's fine. The last thing I want to talk about is this simple experiment where I'm taking the landscape of recombination and keeping the average recombination rate across the chromosome the same, but I'm introducing this dip in the middle that has a lower rate of recombination. And I don't know if any of you have ever done forward simulation before, but it's pretty computationally intensive, and I submitted these jobs to our HPC last week, and I was like, surely they'll start in time for me to get results for my conference presentation. But they didn't. They're still stuck in the queue as we speak. So unfortunately, I don't have any results to share with you about recombination, but they should be coming soon. So stay tuned, I guess. So that's kind of unfortunate, but that's where I'm at. So conclusion slide. The SLIM forward simulation framework is recapturing the past work on turnovers, which is nice. Everything's working. And I think the landscape of recombination is potentially a powerful force that shapes the rate of these sex chromosome turnovers across different clades. So this is very much ongoing work. There's a lot of things that I want to do to build on these simulations. I want to include interactions between deleterious and sexually antagonistic mutations under a wider parameter range, so I can see how the different models sort of interact with each other in a more complex setting. More realistic landscapes of recombination that are informed by empirical landscapes and can vary between the sexes. And then I'm also interested in the effect of these landscapes on the origins of sex chromosomes in the first place and not just in their turnover. So that's in the more distant future. So with that, I'd like to thank the members of my lab and my funding sources. And shameless plug, I'm starting my lab at the University of Rochester next year, and I'm looking for students to start in the fall of 2025. We're going to be working on phylogenomics pretty broadly using primarily theoretical and computational approaches. So if that sounds exciting to you, please get in touch with me. I'd be happy to chat, and I'll be happy to take any questions.